Hello to all. Today we will be going to discuss about the double fertilization, which is a unique and a universal feature found in the angiosperm. No other category of the plants are having such type of the double fertilization. The double fertilization was first shown by a scientist whose name was Navashin in two plants which were named as Fritillaria and the Lilium. Now you must know that in angiosperm, where do the double fertilization occur? So we know very well that in ovary, ovule is present, and in ovule, embryo sac is present, which is the site of the double fertilization, and this embryo sac is also called as female gametophyte. Okay, and the ploidy level of the embryo sac is N, means it is haploid. Okay, now we know very well that this is the carpel. This is the carpel, and the swollen part of the carpel is called as the ovary. And in ovary, what is found? In ovary, ovule is found. This is the ovule, and in ovule, here at the center, what is found? Embryo sac is found, and here the double fertilization occur. So in ovary, ovule is present, in ovule, embryo sac present, and embryo sac is the site of double fertilization. So very first we must be knowing about that what is the embryo sac. So this is an embryo sac, and in maximum angiosperm, the embryo sac is 7-celled and 8 nucleated. 7-celled and 8 nucleated. So we will be counting the 7 cells and the 8 nucleus. Say for at the micropylar end of the ovule, the embryo sac is having 3 cells. Okay, this is the egg cell, which is acting as female gamete, and its ploidy level is N, its number is 1. Okay, and these two are the synergids, right, and their ploidy level is also N, and one egg cell and 2 synergids are together called as egg apparatus. Together they are called as the egg apparatus. Okay, and in synergids certain finger-like projections are present, and these finger-like projections are called as filiform apparatus. In synergids, finger-like projections are present, called as the filiform apparatus. This filiform apparatus release certain chemotropic substances. Which attract pollen tube towards synergids, and like this, through the synergids, the pollen tube will enter into the embryo sac. Okay, now at the chalazal end, 3 more cells are present, and these 3 cells are called as the antipodal cells. They are also having the ploidy level of N, and they are 3 in number. Okay, so we have counted 3 antipodal cells, 1 egg cell and 2 synergids. So 3, 4 and 2, 6. Now total there are 7 cells. So we have to count one more cell. At the center is present the central cell. This is called as the central cell, right? And it is 1 in number. And at the center, 2 nuclei are present. These two nuclei are called as polar nuclei. Each polar nuclei is having the ploidy level of n. So there are two polar nuclei, so n plus n. But just before the fertilization, these polar nuclei combine together and they form a secondary nucleus. And because the secondary nucleus is formed by the fusion of the two polar nuclei, and hence its ploidy level is 2n. But always remember that just before the fertilization, secondary nucleus is formed, otherwise the polar nuclei are present. So altogether, see that we have counted the 7 cells and now we have to count the 8 nuclei. So 8 nuclei, how the 8 nuclei there? 1, 2, 3, 4, 5, 6, 7 and 8. So altogether there are 7 cells and 8 nucleus in the embryo sac, and this embryo sac is acting as the site of the double fertilization. Okay. Now what happens that we know that the pollen grain, when reaches the stigma surface, it germinates. Okay. The pollen grain, when reaches the stigma surface, it germinates like this type. This is the germination of the pollen grain, and whenever the pollen grain germinates, a pollen tube rises. This is a pollen tube, and the contents found in the pollen grain are now released in the pollen tube. A pollen tube is having one tube nucleus at the tip, and also it consists of two non-motile male gametes. Okay, so a germinated pollen grain consists of two non-motile male gametes and tube nucleus. To be remembered here is that the tube nucleus will never participate in the double fertilization. Only in the double fertilization these two non-motile male gametes will be participating, and these male gametes are carried inside the embryo sac through the pollen tube, and when pollen tube carries the male gametes to their destination, it is called as siphonogamy. It is called as siphonogamy. Okay, so this is the embryo sac and this is a germinated pollen grain consisting of one tube nucleus and two non-motile male gametes. Now we will be trying to understand that how do the double fertilization occur in the embryo sac. Now what happens that at the time of the fertilization, the pollen tube move towards the ovule, which is found inside the ovary. Okay, so first of all, pollen tube moves towards ovule and enter ovule through their micropylar end. Means the pollen tube generally enters inside the ovule through the micropylar end. Okay, and there is a ovular structure known as obturator. There is a ovular structure known as obturator guides the pollen tube. So that the pollen tube enters into the ovule through the micropylar end. This obturator is an ovular structure. It is made up by the funicle or the placenta. Okay, and it releases certain chemical substances due to which the pollen tube is attracted towards the micropylar end and the pollen tube enters the ovule. Now, from ovule, the pollen tube enters embryo sac. Now, how it enters the embryo sac? We know very well that in synergids, finger-like projections present. Finger-like projections present called filiform apparatus, which release chemotropic substances, due to which pollen tube is attracted towards synergids. Pollen tube is attracted towards synergids, and now pollen tube enters embryo sac through any one of the synergid. Okay. Now, as soon as it enters into one of the synergid, so what will happen? The synergid disintegrate. And the contents of the pollen tube are released in cytoplasm of synergid by a subterminal pore present in pollen tube. Okay. From ovule, the pollen tube enters the embryo sac. In synergids, certain finger-like projections are present called as filiform apparatus, which release chemotropic substances, due to which pollen tube is attracted towards the synergids, and now the pollen tube enters embryo sac through any one of the synergids. The synergids disintegrate and the contents of the pollen tube are released in the cytoplasm of the synergid by a subterminal pore present in the pollen tube. Means just before the tip of the pollen tube, a subterminal pore is present, and by that subterminal pore, the contents of the pollen tube are released in the embryo sac. Okay. Now, one thing to be remembered here is that always remember, pollen tube never move beyond the level of synergids. Okay. Pollen tube never moves beyond the level of the synergids. Whatever the contents are released, that are released at the synergid level only. Beyond the synergids, the pollen tube will never move. Okay. And we know very well that there are two components found in the pollen tube, right? Means one tube nucleus and two male gametes are now released in embryo sac. Okay. Now what happens, that the tube nucleus soon disintegrate. As I have said earlier also, that the tube nucleus will not participate in the double fertilization. So this tube nucleus will disintegrate. Okay. Now there are remaining 2 male gametes. Now how they will fuse? So see here that now the double fertilization will occur in the embryo sac. And what is double fertilization? In double fertilization, syngamy and triple fusion occurs. In double fertilization, syngamy and triple fusion will occur. We can also say that double fertilization means fertilization occur twice in embryo sac, and in this double fertilization 2 important acts occur. One is known as the syngamy and another is known as the triple fusion. Now one thing to be noticed here is that many times this question is asked that in double fertilization how many nuclei participate. So always remember, in double fertilization total 5 nuclei participate. In couple of minutes I will be counting the 5 nuclei. Okay, in double fertilization 5 nuclei participate and 3 gametes also participate. So always remember, when double fertilization occur in embryo sac, total 5 nuclei and 3 gametes participate. Now what is syngamy and what is triple fusion? See here, we know very well that in the embryo sac 2 male gametes were released. So the first male gamete out of the 2 male gametes, the first male gamete, which is haploid, will fuse with the egg cell. Now egg cell is also having the ploidy level of N, and this egg cell is acting as a female gamete. Now we know very well that this is a male gamete and this is a female gamete, and fusion of male and female gamete is called as syngamy. Means N plus N, fusion of male and female gamete is called as syngamy. So the result of the syngamy is the formation of the zygote. Now in the embryo sac, zygote is formed, and this zygote will be having the ploidy level of 2n because it is formed by the syngamy. N plus N is equal to 2n. So now what is formed? Zygote is formed. Now this is the first fertilization happened, but we know very well in double fertilization occurred twice. So now the second male gamete, which is also having the ploidy level of N, fuses with the secondary nucleus, and secondary nucleus is having the ploidy level of 2n, because we know very well secondary nucleus was formed by the fusion of the 2 polar nuclei. Just before the fertilization in the embryo sac, the 2 polar nuclei fuse together to form the secondary nucleus. So this fusion of the second male gamete with the secondary nucleus is called as triple fusion. Means N plus 2n, and due to which what will happen? A 3n structure will be formed, and that is known as pen. That is known as the 3n. This pen is called as primary endosperm nucleus. This is called as primary endosperm nucleus. Okay. So this is a 3n structure. Later on what will happen? That in embryo sac, the zygote will be converted into embryo. And this pen will be converted into primary endosperm, or we can say it will be forming this is the primary endosperm nucleus will form the endosperm. and this endosperm will also be having the ploidy level of 3n and embryo will be having the ploidy level of 2n. And this endosperm is a nutritive tissue. This is a nutritive tissue and it gives nutrition to the developing embryo. It provides nutrition to the developing embryo. So these are the post-fertilization changes. Okay, that zygote will be converted into embryo and pen will be converted into endosperm, and endosperm will provide nutrition to the embryo. Okay, now one thing to be remembered is that after double fertilization, the central cell of embryo sac is called PEC, that is primary endosperm cell. This is called as primary endosperm cell. So what happens now? See here, after the fertilization you can see the embryo sac, that it seems to be like this type. That is, the synergids disintegrate, these synergids will disintegrate, these synergids will disintegrate. Okay, here the egg cell was there which has been fused with the male gamete to form the zygote. This is 2m. Here antipodal cell also disintegrate, and here at the center what is formed? A 3n structure is formed known as the pen, right? And the central cell is now called as PEC, which is also called as the primary endosperm cell. Okay, so the antipodal cell degenerate, the synergids degenerate, means after fertilization only two components are present. One is the pen and another is known as the zygote. This pen, which is known as the primary endosperm nucleus, will later form the endosperm, and this endosperm is a nutritive tissue which will give nutrition to the developing embryo, and this zygote which will form the embryo later. Okay, so this was the double fertilization, and at last you can see that in double fertilization how many nuclei have been participated? 5 nuclei have been participated. How? One nuclei of this male gamete, another nuclei of this male gamete, okay, 2, third nuclei of the female gamete 3, and 2 polar nuclei of the secondary nucleus. So altogether 5 nuclei and 3 gametes. 3 gametes how? 2 male gametes and 1 female gamete. Okay, so like this in double fertilization 5 nuclei and 3 gametes participate. Okay, so this was all about the double fertilization, which was the unique feature found in the angiosperm. Keep watching my videos. We will be coming soon with some few new videos. Thanks a lot.